On today's Bio5B crash course, courtesy of your supplemental instruction leaders, we will be talking about immunity and immune systems. This was a very dense lecture, so I'm going to try my best to cover as much ground as possible in a short amount of time. So let's begin. Just a reminder that an immune system is all systems that defend an organ against disease. As discussed in class, we have two types of immunity, innate and adaptive. I think this diagram does a good job at simplifying the main differences between innate and adaptive immunity, but we are going to explore these differences a little deeper in the next few slides. Let's start with innate immunity. Innate immunity is the recognition of traits shared by broad ranges of pathogens using a small set of receptors. It is also very fast and occurs in all animals. Innate immunity includes barrier defenses such as skin and exoskeletons that are physical barriers to pathogen entry. Mucous membranes are also barrier defenses that create fluid that trap and wash away pathogens. We also have phagocytes that engulf pathogens in an amoeba-like fashion. There are two types of phagocytes. There are neutrophils that are present in blood vessels, and there are macrophages, which are present in all tissue. Additionally, dendritic cells are another part of innate immunity and are responsible for engulfing pathogens, breaking them up, and presenting parts of the pathogens, called antigens, on their cell membranes. This will activate the adaptive immune system, which we will talk about later. Antimicrobial peptides directly destroy and disrupt pathogens, and they do this by disrupting membrane integrity in bacteria. Interferons are additional antimicrobial peptides that are secreted by virus-infected cells to warn other nearby cells of viruses. They are secreted by host cells and activate inhibitors of viral replication in neighbor cells. Lastly, we have natural killer cells that detect abnormal proteins from infected or, say, cancer cells and dock to the problematic cell. The natural killer cells will release proteins that form pores in the cell membranes, allowing water in and bursting and destroying the target. Inflammation is also part of innate immunity and is triggered by physical injury and infection. This response involves mast cells that release histamine, which attract neutrophils that widen capillaries, resulting in the inflammation. Here are some visuals for a few examples that we talked about. Here we have our dendritic cells, our phagocytes, the natural killer cells, and the inflammation response. So now let's move on to adaptive immunity. In comparison to innate immunity, adaptive immunity is much slower, only occurs in vertebrates, and acts over a longer time frame than innate immunity. What is so special about adaptive immunity is the highly specific reception that occurs. This high specificity of receptors is generated by lymphocytes. Lymphocytes are our white blood cells, and there are two different classes of lymphocytes. We have T cells, which migrate to the thymus for storage and maintenance, and B cells, which stay in bone marrow for storage and maintenance. These cells express unique receptors called antibodies that bind to a unique part of the pathogen called an antigen. So let's take a closer look at the structure of an antibody on a T and B cell. On the left, we have an antibody of a B cell, and on the right, we have an antibody of a T cell. Both contain variable and constant regions. The variable region is different in each antibody and gives the antibody a unique antigen binding ability. The constant region, on the other hand, is the same in all antibodies. It is important to remember that antibodies are highly specific and normally just bind to one part of one molecule from a single species of pathogen. We must also remember that every B or T cell will only produce one type of antibody, but every antibody is unique. Okay, so now let's talk about how these antibodies bind to an antigen and produce an adaptive immune response. This adaptive immune response begins with the binding of an antibody to its matching antigen. This leads to cell division and increased production of more of this type of B cell/antibody type. There is also massive production of the soluble form of this type of antibody via plasma cells and formation of long-lived memory cells that can be made quickly if reinfection occurs. These memory cells allow antibodies to quickly be reproduced if reinfected with the same pathogen. This is essential for vaccines, which stimulate the immune system to generate long-lived memory cells for that particular pathogen. So one last point I would like to cover is how antigens kill the pathogens, and there are multiple ways in which they do this. The first is neutralization, in which antibodies flood the surface of the pathogen so it cannot interact with cells in the host. Another killing method is opsonization, which makes pathogens more easily found by phagocytes. Lastly, we have recruitment, in which antibodies bring antigen to helper T cells that mediate additional pathogen-killing pathways. This may lead to humoral immunity, which is secretion of antibodies into blood plasma to fight pathogens in body fluids, or cell-mediated immunity, which refers to attack of host cells that have already been infected. That is basically all for today's video. Hopefully it was helpful in providing a brief overview of immune systems. If you have any more questions, please come see us in office hours or email us. Have a good weekend and good luck on your midterm.